Here's a fact that shows up on almost every exam. Your innate immune system reacts exactly the same way every single time it meets a pathogen, first exposure or 50th. It never gets faster. Adaptive immunity is what changes that. In this video, we'll cover innate immunity, adaptive immunity, how they stack up side by side, how the two arms talk to each other, and the full timeline of an infection. I'll flag the timestamps in the description below if you want to jump straight to one. But before we split innate and adaptive apart, you need to understand why the immune system is built as two arms in the first place, and that starts with a handful of core principles every single question on this topic is built on. The immune system works like an integrated surveillance network. It has to constantly detect and eliminate non-self antigens, disease cells, and damaged tissue. So the first job of the whole system is distinguishing self from non-self. The second job is memory, the ability to amplify its response if it sees the same threat again. And the third job is knowing when to stop, returning to homeostasis once the pathogen is cleared, because a system that never shuts off starts attacking its own tissue. All of this runs on diverse receptors and specialized effector functions, held in check by self-limiting signals that prevent autoimmunity and malignancy. And that sets up the single most important distinction in this whole topic. Innate immunity is your first responder, rapid, fixed, and broad. Adaptive immunity is slower, but it's specific, and it remembers. Now that we've covered the core principles, let's talk about the first arm, innate immunity. This is your immediate, non-specific defense. It's evolutionarily ancient, and it doesn't require any prior exposure to work. It starts with physical and chemical barriers, intact skin, mucous membranes, your normal flora, acidic pH, lysozyme, antimicrobial peptides, even temperature. These stop most pathogens before they ever get inside. On top of those barriers, you've got humoral factors circulating in the blood, the complement system, acute phase proteins, and cytokines that recruit inflammatory cells to the site. And finally, the cellular effectors, phagocytes like macrophages, neutrophils, and dendritic cells, granulocytes like basophils and eosinophils, and natural killer cells that patrol for infected or abnormal cells. Put all of that together, and you get a system that's ancient, immediate, and nonspecific. It reacts to categories of danger, not individual pathogens. So, what actually makes innate immunity work the way it does? It comes down to a handful of key characteristics, and this is exactly where exam questions like to test you. First, the receptors are germline encoded. These are pattern recognition receptors, or PRRs, and they recognize pathogen-associated molecular patterns and damage-associated molecular patterns, PAMPs and DAMPs. Because these receptors are fixed from birth, diversity is limited. There are only about 100 PRRs, compared to millions of possible adaptive receptors. And because nothing changes between encounters, there's no memory. Every exposure triggers the same intensity of response. What you do get in exchange is speed. Activation happens within minutes to hours. And here's the USMLE focus box. PAMPs include LPS from gram-negative bacteria, lipoteichoic acid from gram-positive bacteria, flagellin, and viral dsRNA. DAMPs, the ones released from your own injured cells, include uric acid, ADP, and HMGB1. With that in mind, now let's move to the second arm, adaptive immunity. The central players here are B and T lymphocytes, and their receptors aren't fixed like PRRs. They're generated by somatic recombination. That process is what gives adaptive immunity its defining hallmarks. Specificity means each lymphocyte carries a single unique receptor for one particular antigen. And because that receptor is built through recombination rather than inherited fixed, diversity is enormous, somewhere between 10 to the 9th and 10 to the 11th possible specificities. Which is exactly why memory exists here and not in innate immunity. On re-exposure, the response is faster, stronger, and more effective. None of that works, though, Without self-tolerance, central and peripheral mechanisms that stop these powerful receptors from attacking your own tissue, and self-limitation, where effector cells contract through apoptosis once the antigen is cleared, so the response doesn't run forever. So, where does that memory actually come from mechanistically? It starts with B cells, which differentiate into plasma cells that secrete antibodies. Those antibodies work through opsonization, neutralization, complement fixation, and antibody-dependent cellular cytotoxicity. At the same time, CD4-positive T helper cells are secreting cytokines, and this is what activates macrophages, drives B cells toward antibody production, and helps cytotoxic T cells do their job. Then you've got CD8-positive cytotoxic T cells, which kill infected or malignant cells directly through two mechanisms: perforin and granzyme, which punch into the target cell, and the Fos/Fosl pathway, which triggers apoptosis from the outside. And this is exactly why immunological memory is the basis of vaccination. Secondary responses are carried by long-lived memory T and B cells, and by plasma cells that are already primed to pump out high-affinity antibodies. Let's move to the comparison and put everything you just learned side by side, because this table is exactly how these two arms get tested against each other. On specificity, innate reacts to shared structures, PAMPs and DAMPs, while adaptive targets specific antigen epitopes. On diversity, innate is limited to around 100 PRRs. Adaptive is extremely high, thanks to somatic recombination. And here's the row that matters most: memory. Innate has none. Adaptive gives you an enhanced secondary response. That's the whole reason vaccines work. Time to activation. Innate responds within minutes to hours. Adaptive takes days on the primary exposure, but drops to hours once memory is established. Components. Innate relies on barriers, complement, phagocytes, and NK cells. Adaptive relies on B cells, T cells, antibodies, and the lymphoid organs that house them. Both arms have self-tolerance, though adaptive's is far more tightly regulated through thymic selection, anergy, and regulatory T cells. And both arms are self-limiting. Innate resolves its inflammation. Adaptive contracts after the antigen clears. Quick check before we move on. Based on everything we just compared, do you remember which arm of immunity has no memory at all? Innate or adaptive? You just covered this a minute ago, so take a second and drop your answer in the comments. I'll keep building on it as we go. So where does that leave us? These two arms aren't operating in isolation. They're locked in a positive feedback loop, and this pathway is exactly how that loop starts. It begins with a pathogen. That pathogen is recognized by a PRR. The cell that recognized it then phagocytoses the pathogen and processes its antigen. That antigen-presenting cell then presents the processed antigen to a T cell. Alongside that presentation, cytokines and co-stimulatory signals are released. And that combination, antigen presentation plus cytokines and co-stimulation, is what finally activates T and B cells. In other words, innate immunity isn't just a first responder, it's the trigger that switches adaptive immunity on. Let's break that loop down into its actual bridges. First, the phagocytes, dendritic cells and macrophages, engulf pathogens and present antigen via MHC directly to T cells. Because of that presentation, inflammatory cytokines get released, IL-1, IL-6, TNF, and IL-12, and these cytokines are what shape which direction adaptive immunity differentiates toward: Th1, Th2, or Th17. Here's the catch, though. This loop runs both directions. Antibodies, once adaptive immunity produces them, opsonize pathogens, boost phagocytosis, and activate complement through the classical pathway. And CD40L, along with other co-stimulatory signals, bridges the two arms directly. Which is exactly the positive feedback loop this whole section is built on. Innate signals switch adaptive cells on, and adaptive's own effector molecules, cytokines and antibodies, come back around and boost innate killing and inflammation. Next, we need to look at how all of this unfolds over the actual course of an infection, because timing is exactly how examiners test whether you understand innate versus adaptive. It starts with the innate response in the first 0 to 4 hours, driving inflammation and phagocytosis. Then, from around day 2 to day 7, adaptive priming begins. From day 7 to day 14, you get the full adaptive effector response, and after that, resolution and memory formation. In other words, the innate response peaks within hours, but if the pathogen persists, adaptive immunity doesn't fully engage until about a week in. Which is exactly why memory matters so much. Once memory cells exist, that same week-long delay collapses down to just 1 to 3 days on re-exposure. So, where does all of this land on your exam? Let's go through the clinical pearls one at a time, starting with memory, the hallmark of adaptive immunity. On secondary exposure, you get rapid expansion of memory B and T cells, plus high-affinity antibodies already primed to respond. Next, PAMPs and PRRs, specifically toll-like receptors or TLRs. TLR4 recognizes LPS. TLR3 recognizes double-stranded RNA. TLR9 recognizes CpG DNA. These three come up constantly on exams, so lock them in. Which brings us to what happens when self-tolerance fails. Autoimmune disease, like lupus or rheumatoid arthritis. Central tolerance is where this gets stopped in the first place, in the thymus for T cells and in the bone marrow for B cells. Building on that, immunodeficiency can hit either arm. On the innate side, think chronic granulomatous disease and complement deficiencies. On the adaptive side, think SCID and HIV. Which is exactly why vaccines work the way they do. They exploit adaptive memory to give you long-term protection without ever causing the actual disease. And that brings us right back to the exam tip that opened this whole video. Innate immunity does not improve with repeat exposure. Re-exposure to the same pathogen produces a quicker, more robust response only in adaptive immunity, because that's the arm with memory lymphocytes doing the work. So where does that leave us? Innate immunity is fast, fixed, and never learns. Adaptive immunity is slower but specific, and it remembers. Together, they form one integrated surveillance network, and that's host defense. Thanks for watching.